Hello, welcome to Medical Immunology. Today we'll be focusing on T cells and specifically focusing on cytokines and different T cell and immune cell subsets. Lecture objectives is we'll begin focusing on CD8 positive cytotoxic T cells. From there, we'll move on to different subsets of CD4 positive T cells. And then we'll go into regulatory T cells and their function and mechanisms. So T cells are the most important cell of the immune system. They really orchestrate the entire immune response and the selectivity and ability to kill different pathogens. Because we are exposed to so many different types of pathogens, from viruses to microbes that persist in macrophage vesicles, to helminth parasites and different types of fungi, is that we have a variety of different T cell subsets that are very focused on killing each of these different types of microbes. So in order to kill the pathogens, we need to have very specialized T cells that have great abilities to carry out effector functions, but spare normal and healthy tissues. So as such is that there are special T cell subsets that are designed to kill each type of pathogen. And that's what we'll be reviewing today, going through each one of these one by one to show how they work and why they work. So to begin, we're going to talk about cytotoxic CD8 T lymphocytes. So CD8 T cells are a type of T cell that arise in the thymus. And these have specificity towards MHC class 1. So specifically, these will recognize viral antigens presented in the context of MHC class 1. So CD8 cytotoxic lymphocytes mediate direct killing of virally infected cells. So in this case, the goal of these cells is to identify cells that are infected with virus and then selectively kill the virally infected cells. So they aren't directly killing the virus themselves, but they're killing the cells that have been infected with the virus. And the idea there is that these cells that are infected with the virus have essentially been converted into virus-producing factories. And so if you can kill these cells that are infected with virus before the virus kills the cells themselves, that this will allow you to stop viral production in its tracks within the body. So the cytotoxic T lymphocytes, they will bind to cells that express MHC class 1 that is presenting viral antigen. And using that CD8 molecule to stabilize this interaction, they will bind and then kill that target cell. So they will induce apoptosis or cell death in their targets. From there, they will then migrate to a new target, a new cell, a different cell that also expresses that same MHC class 1 that's presenting that viral antigen, and then go from there and kill each consecutive cell that is infected with the virus. And then once it no longer finds any virally infected cells, it'll go off and keep searching for more virally infected cells. So the cytotoxic T lymphocytes recognize viral antigen complex with MHC class 1 using the T cell receptor, and then it can then be recycled to kill multiple target cells using its cytotoxic effector molecules. So what are these cytotoxic effector molecules? These are things like perforin, granzymes, granulysin. These are things that it secretes out and uses these to kill the virally infected cells. So cytotoxic T lymphocytes, CD8 positive cytotoxic T lymphocytes, are very effective at killing. And one of the reasons they're so much more effective at killing than other cells of the immune system is because they express these unique molecules. So these are proteins that are in the granules of cytotoxic T cells, and perforin will perforate the host cell, which will aid in the delivering of contents into the cytoplasm of the target cells. Granzymes are specific enzymes, proteases, that will then cleave different molecules inside the host cell that will then drive apoptosis. And then granulysin will also help to contribute to induce apoptosis. And then additionally, they will also express a variety of other pro-inflammatory cytokines. These could be protein cytokines like interferon or tumor necrosis factor, or these could also be pro-inflammatory lipids like leukotrienes. So cytotoxic T lymphocytes use their T cell receptors to specifically recognize viral antigen presented in the context of MHC class 1. So what this means is that they can specifically recognize infected cells, cells that are infected with virus, and then it can select that cell for death, and then the neighboring cells that are presenting self-antigen and not viral antigen in the context of MHC class 1 are spared. So in this case, cells that are not presenting viral antigen in the context of MHC class 1 will be spared the effects of that cell death because they will not be recognized by the T cell receptor of the CD8 positive cytotoxic T lymphocyte. So all the cells in a tissue are susceptible to killing. However, only the infected cells are killed, and that's because of specific recognition of the viral antigen, and then also polarized release of those cytotoxic granules containing the granulysin and the perforin. So these will be just, selectively released into that infected cell that will kill the infected cell and leaving those neighboring bystander cells unaffected. So why do we need to know all of this? So this is one of the first things that was looked for in the initial phases of the coronavirus response. It's when the coronavirus pandemic was just beginning in early 2020. One of the things they looked for in peripheral blood was to look at CD8 cells and understand, do these cells exist? And if they do exist, what are they expressing? So this is a flow cytometry plot. So in here you can see that this is a flow cytometry plot gated on CD8 positive T cells. And all of these cells express a lot of perforin, a lot of granulysin as well. And so specifically, this is a case study of a 50-year-old man who presented early in the pandemic in January with symptoms consistent with COVID-19. Diagnostic tests were consistent with COVID-19 as well. And then on day 14 of his illness, he developed hypoxemia, so low blood oxygen, shortness of breath, and then had a sudden cardiac arrest. So peripheral blood, upon examination, they found that the counts of CD4 and CD8 T cells were reduced. However, although they were reduced, the cells that were still there were hyperactivated. And by hyperactivated, they mean lots of perforin, lots of granulysin. So this COVID-19 ARDS, acute respiratory distress syndrome, coincided with overactivation of these CD8 T cells, and that was correlating with the severe immune injury in those patients. So we've talked about activation of CD4 T cells. So past few slides, we've been focused on CD8 positive killer T cells, cytotoxic T cells. Now we're going to get into CD4 positive helper T cells. So we previously discussed the first activation signal presented by MHC class 2 and the T cell receptor, and then the co-stimulatory signal mediated by B7 on the antigen presenting cell and CD28 on the T cell. And then we didn't really talk a lot about this signal 3. But this signal 3 is so important because this signal 3 is going to mediate the specificity of these activation steps. So when these cells get activated, how are they getting activated? And what I mean by that is that they're going to be differentiating and turning on different transcriptional programs that are designed to kill specific things. And so these specific things that they're going to kill are guided by these cytokines, this signal 3. And then importantly is that these signal 3 targets, these are very good drug targets. So large portion of the immune acting drugs that are commonly prescribed these days for a variety of chronic inflammatory and myeloproliferative diseases are targeting this signal 3 pathway. So it's important to understand the signal 3 because this is going to keep coming back first off in this lecture when we're talking about blocking the signaling using different kinase inhibitors to suppress the immune system. But we're also going to be bringing this back later when we talk about autoimmune disease and the different drug targets appropriate for various autoimmune diseases. So signals 1 and 2 will work together to activate NFAT and NF-kappa B in the CD4 positive T cell. However, signal 3 is going to be acting differently. That will support NFAT and NF-kappa B. So you'll be activating different transcription factors, and these different transcription factors will help to guide the T cell to make specific effector molecules that are better at killing intracellular bacteria or extracellular bacteria or fungi or things like worms and other parasites. So we've heard of these different TH subsets. So these are your different helper T cell subsets. H stands for helper, T stands for T cell. So we have TH1, TH2, TH17, and on, follicular helper cells, regulatory T cells. All of these are guided by this signal 3. So each of these subsets, the TH1, these are very good at killing microbes that persist in macrophage vesicles. So all of these will be coming back the more you learn about microbiology, is that this cell type, TH1, is going to be really good at helping turbocharge our macrophages to kill intracellular bacteria. TH2 cells are really good at turbocharging our allergic cells, things like mast cells, things like mucus-secreting cells, in order to kill extracellular parasites. The TH17 cells are very good at killing extracellular bacteria and fungi by activating the local epithelium, as well as activating neutrophils and really turbocharging these neutrophils to kill these extracellular bacteria and fungi. Follicular helper cells are special T cells that are really good at helping out B cells and getting those B cells nice and activated to produce lots of mature antibodies. And then regulatory T cells are going to be our negative regulator of this entire response. The regulatory T cells will be shutting down the immune system. So how does this stuff work? We have all these different T cell subsets, TH1, TH2, TH17, follicular helper cells, regulatory T cells. How do all these stuff work? How do they get turned on? Is that signal 3. That signal 3 is made by the dendritic cells. So if we go back and just understand and recap what we know about dendritic cells. So dendritic cells are activated in the periphery. Dendritic cells are activated in the local tissues. They then migrate to the lymph node. And in the lymph node, they can begin interacting with T cells. In the lymph node, they're going to be interacting with these T cells, and they're going to be turning on these T cells in a way that's telling the T cell, hey, this is what we're dealing with. So one of the ways it does that is by presenting antigen, getting antigen-specific T cells ready to go out and fight that specific bug. But another way it's going to do that is they're going to secrete out these different fate-specifying cytokines. So all of these are going to be made by that dendritic cell. And what they're going to do is they're going to tell that dendritic cell, hey, this is what we're dealing with. We're dealing with intracellular bacteria. So you need to start making a lot of interferon in order to go out and help turbocharge our macrophages to kill those intracellular bacteria. Or we're dealing with a worm, we're dealing with a parasite. You need to go out and start making a lot of IL-4, IL-5, so we can start dealing with that parasite. In addition, it could say, hey, we have a lot of extracellular bacteria or fungi, so you need to go out and start making a lot of IL-17 to turbocharge our epithelium, to activate our neutrophils, to go out and kill that specific thing. Making a lot of IL-6, say, hey, it's time to make a lot of antibodies so that we can go out and help neutralize all the bugs that are in the that we need to fight. Or saying that, hey, things are really super activated right now. We need to go out there and start calming things down with these regulatory T cell mechanisms. So up here we have the subset inducing cytokine. And then these are the cytokines that are made by that specific subset, and then the subset name down here. So interferon and IL-12 will work to activate cells to make more interferon. IL-4 will work to make cells that produce more IL-4 and IL-5. These three cytokines together here will work together to turn on this pro-inflammatory cell that's making a lot of this IL-17. IL-6 will turn on the cells to make this type of cytokine here that's going to help turbocharge our B cells. And then these two cytokines will work together to make these regulatory T cells that make these anti-inflammatory cytokines that will shut down the whole system. So in order for this to work is that these cytokines have to turn on transcriptional patterns inside the T cell. So what we have here is that we've learned about transcription factors in T cells. What I mean by that is that transcription factors are molecules that can recruit an enzyme called RNA polymerase to specific regions of the nucleus called promoter regions, and this can then turn on inflammatory processes in T cells. But it's not just NFAT, it's not just NF-kappa B, but we also have these other transcription factors called STATs. These STATs are named for signal transducers and activators of transcription. These STATs are all different. These STATs are all things that are involved in activation of different patterns inside the T cell. So for example, activation of STATs will help turn on other types of transcription factors like TBET, and then this TBET will really turbocharge that T cell to make a lot of interferon. STAT6 will turn on other things like GATA3, and then GATA3 will help that T cell make a lot of interleukin-4. More gamma is going to be turned on by STAT3. So basically, all these cytokines are going to be working to turn on different transcriptional patterns. And then these transcriptional patterns will make lots and lots of cytokines. So here, for all of our different subsets, what constitutes a subset? A subset is dictated by, first, the transcription factors that are active in that subset, and second, the cytokines that are made by those specific things. And the idea is that if we're infected with a worm, we don't want to make a bunch of cytokines that aren't good at killing worms. We want to make cytokines that are really good at helping kill those worms. So the way this stuff works is that when cytokines are released by dendritic cells or any cell that's making cytokines, these cytokines are released into the environment. And the environment, the extracellular space, these cytokines are now free to go out and bind cytokine receptors. So when cytokines bind cytokine receptors, this is then going to cause changes in the cells that express those receptors. So for example, if I'm a T cell and I'm looking and trying to understand what the dendritic cell is telling me, I see this activated dendritic cell. I know he's activated because he's expressing MHC class 2. He's expressing these co-stimulatory molecules like B7. I'm bound to that dendritic cell. I'm sensing it. We're signaling, we're talking, but I want to know what that dendritic cell is telling me. One of the ways I can understand what the dendritic cell is telling me is by listening to the cytokines that he's making. I have all these cytokine receptors on my surface, but the cytokine receptors that are getting engaged by the cytokines made by that dendritic cell, now I start to understand what that dendritic cell is saying. Okay, so he's making these cytokines, they're binding to my certain cytokine receptors, and that's going to turn on kinases. These are types of kinases called Janus kinases. What the Janus kinases are going to do is they're going to take and phosphorylate transcription factors. These are my transcription factors called STATs. STATs are an additional type of transcription factor that work with NF-kappa B, that work with NFAT in T cells. And what that's going to do is I'm going to phosphorylate my STATs, send them to the nucleus, and then start producing a lot of new gene transcription. So again, why is this important? Is again, signal 1, signal 2, signal 3. We know we can drug signal 1 and signal 2 with cyclosporin, things like calcineurin inhibitors, cyclosporin, things like that, tacrolimus. We also know we can block NF-kappa B with things like cortisol derivatives, things like steroids, corticosteroids, dexamethasone, prednisone, all of those, hydrocortisone, all of those are really good at blocking signals 1 and signals 2. But what about signals 3? We can also block signal 3 using different drugs. And these drugs are called Janus kinase inhibitors, JAK inhibitors. So many of the commercials that you'll see on your football or NBA games, a lot of these are commercials for Janus kinase inhibitors. The big ones are really going to be your Rinvoq and Xeljanz, tofacitinib in particular. These are approved for a number of inflammatory diseases. A couple of them include ulcerative colitis, psoriasis, rheumatoid arthritis. We'll go into more detail on those later in this class. But those are chronic autoimmune inflammatory diseases. And one way to kind of damp down the inflammation, but without doing a total sledgehammer on the system, is to use JAK inhibitors. And these JAK inhibitors that target JAK3, they also have other activity on other JAKs, but JAK3 is really one of the dominant Janus kinases. These can then block a lot of the activity of those cytokines. We also have other Janus kinase inhibitors approved for myeloproliferative diseases. Those are some bone marrow cancers and things like that. So this is going to be Jakafi, targeting JAK1 approved here, and then for patients that are refractory to JAK1 inhibitors, JAK2 inhibitors can also be used. So these Janus kinases are very important drug targets. These cytokines are very important inflammatory things. So block the cytokines, reduce the inflammation, have beneficial outcomes on a number of inflammatory and myeloproliferative diseases. So this is just another way to look at this. A lot of these slides are just kind of the same information over and over, just looking at different things. So we have our dendritic cell activating the T cell. Depending on the cytokines made by the dendritic cell, it's going to send the T cells down a number of these paths. So if you activate a variety of these different stats, this will help push the T cell down a number of different lineage states, different subsets, including follicular helper, pro-inflammatory Th1, Th2, Th17. Th1 was the first subset discovered. Th2 was the second subset discovered. Th17 was named because of its signature cytokine, interleukin 17. Interleukin 17 is the most pro-inflammatory cytokine in the body. So interleukin 17 really induces robust proliferation and inflammation. So this very inflammatory cell, very inflammatory cytokine. And then this is the negative regulator. So this is the anti-inflammatory cells. So each of these are different subset-specific transcription factors that'll help to stabilize that subset into making all of those specific secreted cytokines. So differentially activated T cells secrete different cytokines. So we've introduced CD8 positive T cells. Now we're going to go one by one through each of these different classes of T cells. We're going to focus on some of their signature cytokines that they make, what those cytokines do, why they make them, and why it's important for the physiology and the immunology and the ability to kill different microbes. So Th1, the key thing is that it's going to activate macrophages. Th2, the key thing is it's going to activate these kind of barrier systems. So a lot of our allergic cells that are within the different tissues are going to become activated. And then Th17, this is really going to act on the epithelium as well as activating neutrophils. And then Tregs will shut down the whole thing. So just here, you don't need to memorize every single cytokine, but the cytokines that are signature for the subsets are important. So for example, understand that, interferon and tumor necrosis factor are really important for Th1. Know that IL-40, very important for Th2, IL-70, very important, or IL-17, very important for Th17, and then these two, interleukin-10 and TGF-beta, very important for the regulatory cells. And all of this should be on the study guide as well. So what do Th1s do? Th1s will help kill intracellular bacteria. So the way they do that is mainly by activating our macrophages. So they'll produce different cytokines that will turbocharge our macrophages. So this will do things to help engage the macrophages and kill the engulfed bacteria. So these lysosomes that are within the macrophages will become turbocharged once the Th1 cell activates those macrophages. Here we have further activation of these macrophages by the Th1s. In addition, these Th1s make a lot of interleukin-2, which will help activate more T cells. In addition, they'll be secreting out factors that will make their way back to the bone marrow, get the bone marrow to produce more and more and more monocytes. The bone marrow is constantly differentiating blood cells based off of the signals it receives from the body. And so these Th1 cells will be sending signals through the bloodstream back to the bone marrow to tell it that we need more monocytes. It'll help activate the blood vessels, the endothelium, to get more macrophages and monocytes into the tissues. And then it'll help secrete out to these different chemoattractant molecules to get more macrophages and monocytes to the site of infection. And the goal is to kill these intracellular bacteria. Th2s are a little different. So when you have a worm, worms are bigger than intracellular bacteria. As such, you need different things to get them out. So what you're going to start doing is you're going to start making a lot of snot, a lot of mucus. You're going to get a lot of spasming of the smooth muscle to try to expel things out. You're going to activate a lot of your allergic cells to mediate histamine and other things. Things are vasoactive, things that will help you sneeze and have diarrhea to try to get these things out of the system. So you're going to have activation of Th2s that are going to be making a lot of mucus. That mucus from the goblet cells will help prevent adherence of the parasite to the tissues. That IL-13 is going to be acting on the smooth muscle to help expel that worm. You're going to be changing the macrophages to help with the tissue remodeling and repair. You're going to be activating different allergic cells like eosinophils and mast cells, and all of those are going to help release things like interferon or things like histamines. And what those will do is those will all help to mediate allergic responses and to kill those parasites ultimately. IL-17 is going to be working on different effectors. So specifically, it's going to be working on the epithelium. So you're going to get massive hyperplasia in the epithelium. You're going to get massive transcription in the epithelium, where you're going to trigger your epithelium to make lots of antimicrobial peptides. You're going to trigger hyperplasia and a lot of growth of the epithelial cell division and shedding that'll help to impair bacterial colonization. So get a lot of turnover in the skin or in the lining of the gut, depending on where the bacteria is. You're going to get a lot more neutrophils made by the bone marrow because these cells are going to be talking to the bone marrow, using their cytokines, getting the bone marrow to make a lot more neutrophils. And then you're going to be really turbocharging those neutrophils by making lots of chemokines, getting those neutrophils into the site, making a lot of pus, making a lot of neutrophil extracellular traps, recruiting in a lot of other Th17 cells into the tissue. So all of these will be working in concert in order to kill off these extracellular bacteria. So Th17 cells are also very important. Here in this patient that was undergoing our acute respiratory distress syndrome, so that's cytokine storm mediated by his COVID-19 disease. So this is the same patient we saw earlier. This patient had lots of pro-inflammatory CD8 cells, but also lots of Th17 cells. So normally Th17 cells are so inflammatory, you want less of them in the system. Here in this patient, CR7, so this is a chemokine receptor that's very important in activation of Th17 cells. So something characteristic of Th17 cells. You can see this patient had a lot of IL-17 producing Th17 cells. So these are some of the most inflammatory cells in the body. T cells are the most inflammatory cells in the body, but Th17 cells are even beyond that, the most inflammatory subset. And so these Th17 cells are highly pro-inflammatory, and these results suggest that overactivation of T cells manifested by the increase of Th17 and the high cytotoxicity of CD8 cells accounts for the severe acute immune injury in this patient, that cytokine storm. So, this was very important early in the pandemic because we didn't know why people were dying so much. And it was papers like this, looking at these different immune cell subsets that we're covering today, that led people to begin thinking, okay, well, now let's try treating with steroids and then things like that in order to try to mitigate these these effects that we're seeing in these patients. So for our final T cell subset will be our regulatory cells. So our regulatory T cells stop inflammation. So typically in pathologic tissue inflammation, like you might see in autoimmune disease, maybe something like psoriasis, where you have these epithelial cells, our skin. And what might happen is that you might have T cells that are getting activated, reacting against skin epitopes, different keratin or something like that. And so what's going to happen is that all of these inflammatory cells are getting activated by dendritic cells in the lymph node, and they're coming back to the tissue and they're causing pathologic tissue inflammation. So what these regulatory T cells do is these regulatory T cells help with the tissue repair, and they chill out our antigen-presenting cells. They calm down the dendritic cells. They suppress the T cells. They suppress our pro-inflammatory T cells, and this will help lead to tissue homeostasis. So most of the T cells that you see in the gut, most of the T cells that you see in the skin are going to be in this regulatory phenotype where they're going to be preventing inflammation, preventing unwanted inflammation. So these are the cells that help to calm down the inflammation. These are cells that help to turn off all these pro-inflammatory processes that we've been covering earlier in today's lecture. And the way that this works is that first is that in the thymus is that certain types of cells that have certain reactivity for self-antigens, these self-reactive T cells, they'll be deleted out, but a certain portion of them will survive and become regulatory cells, naturally regulatory cells that will go out and go to the tissues and help guard those tissues from unwanted inflammation. Similarly, is that you can have activated cells, and if they're activated in the presence of those cytokines that we covered earlier, things like IL-6 and TGF-beta, what's going to happen is that over time is that those cells can become these induced regulatory cells, and those cells will help by secreting anti-inflammatory cytokines and other mechanisms to help calm things down in the periphery, help shut down the activity of very pro-inflammatory or self-reactive T cells. So this is a type of T cell in purple that can shut down other T cells. And they work to inhibit the activation of other T cell subsets, particularly the Th1s and the Th17s, which tend to be very pro-inflammatory. So how do these things work? First is they express a lot of CTLA-4. We know CTLA-4 is a negative regulator of the immune system. So it makes sense that these cells express these molecules that shut down the immune system. Interleukin-10, TGF-beta, transforming growth factor beta, TGF-beta, IL-10, these are anti-inflammatory cytokines that work to suppress the immune system. They prevent the spread of pro-inflammatory cytokines, so they can bind to pro-inflammatory cytokines and help prevent other cells from seeing them. they can work to block T cell and antigen presenting cells, so they can bind to dendritic cells, and because they're bound to the dendritic cells, other T cells can no longer bind. And then they can directly kill activated T cells as well. So if they find an activated T cell, they can bind to that T cell and kill it. And that can then again prevent that activated T cell from causing unwanted inflammation. So what is an example of immune reaction where you don't want inflammation, where you have something foreign in your body, but you want to protect it? An example of that is in pregnancy. So in pregnancy, that fetus and the placenta have antigens that are both from the mother and the father. And because of this is something that is non-self, that's growing. But what's happening is that in here is that you don't have these pro-inflammatory immune responses in most pregnant mothers, is you don't have that pro-inflammatory response that will be, lead to spontaneous abortion of the fetus. In this case, what you have is that you have an anti-inflammatory immune response. So what that means is that these conceptus-derived antigens are being carried by the uterine vein, by the lymphatic vessel, back to the lymph nodes. And in the lymph nodes is that dendritic cells are picking up these antigens, presenting them to T cells, but they're presenting them in an anti-inflammatory way. How can a dendritic cell present something in an anti-inflammatory way? It really depends on the antigen and how that dendritic cell encountered those antigens. In the case of a bacteria, you have pathogen-associated molecules like toll-like receptor 4 agonists and endotoxin and lipopolysaccharide and flagella that are activating immune receptors, toll-like receptors on the dendritic cell, and that will cause pro-inflammatory activation of the dendritic cell. Here, there's no pathogens, there's no endotoxin derived from the fetus, and as such, the dendritic cell is going to get activated without these toll-like receptors getting engaged, without these pattern recognition receptors getting engaged. So the cytokines that dendritic cell is making are going to be very different than it would make if it saw those antigens in the context of lots of cells dying and lots of microbial products associated with that. So essentially what you're getting is activation of dendritic cells, presentation of foreign conceptus-derived antigens, but it's getting presented in an anti-inflammatory context with anti-inflammatory cytokines being made. And as such is that these T cells are now going to become regulatory cells that can migrate back and help protect that fetus. This is a mouse fetus, but it's a similar concept in humans. So essentially the idea is that these conceptus-derived antigens will expand these regulatory cells, and that will help suppress conventional T cells from activating and then causing inflammatory rejection of the fetus. So these Treg cells dominate the response to the developing fetus. So if you have conceptus-derived antigens activating conventional T cells, these regulatory cells can now help suppress those activated immune cells and then prevent fetal loss from that is secondary to systemic inflammation. You can also have regulatory T cells that are suppressing general T cell activation in general. And it's something that's just fascinating is that in women with multiple sclerosis, for example, that's a chronic relapsing remitting disease of the immune system, so an inflammatory disease, a lot of times those women have better outcomes, so less inflammation when they're pregnant, because these regulatory cells will then dominate the immune response and in order to help reduce the fetal loss secondary to systemic inflammatory processes. So this is going to prevent the activation of pro-inflammatory Th1 and Th17 cells, thus reducing the risk of fetal rejection due to inflammation. There's a lot more reading if you're curious here, but essentially you have these pro-inflammatory immune cells as well as anti-inflammatory immune cells that are good at protecting your own body, but also in the context of pregnancy, also helpful for protecting those. And in the case of cancer, which we'll see in a number of lectures coming up, because these cells are also important for helping protect those cancers from inflammatory rejection as well. So thank you for your time.